Morning, everybody. That didn't sound... good morning, everybody.
Good morning.
Good morning, everybody.
Good morning.
Good morning, everybody.
Good morning.
These guys are much louder. There are more of them. All right, let's try this again. Really loud. Good morning, everybody.
Good morning.
Wow. Good morning, everybody.
Good morning.
All right, hey, it's awesome. Great that you're here. Great that we're here. I cannot begin to tell you how terrific I thought yesterday was. Holy cow. Holy cow. I mean, the people we heard from, the students we heard from, the stories we saw, the stories we heard, the stories we learned were just great. Did you meet some good new people?
Yes.
You promised? Did you meet some good new people?
Yes.
Great. I know Bruce did. Bruce is very outspoken about this. Is this good? This is on the internet. Good morning, everybody who's streaming. All of this is being shared. Would you please tweet this, post this, tell people to tune in, because this is where it's happening now. planetforward.org and all of our hashtags which you have and which are on your programs. This morning is going to be amazing. Today is going to be amazing. You're going to see a presentation in a moment that will pretty much blow you away and talk about science and storytelling. This is going to be it. And then you're going to have a really amazing opportunity out of that. Our town hall that we're going to have here on energy is, frankly, a once-in-a-lifetime opportunity for all of us. The more I think about this and the more I anticipate it, the more excited I am in the kinds of conversation that we're going to have. And then we're going to roll out a whiteboard, and we're going to take your story ideas from that conversation and more, and we're going to write them down, and we're going to imagine and which ones we can pursue, and then we're going to pursue them over the course of the next year. And some of you will help us do that. And then we will give our awards. Who are our Planet Forward StoryFest finalists in the room? Raise your hands. Okay. Okay. What was your story? What's your finalist story?
Solar pump.
Solar, okay. Name and school?
Janet, SUNY ESF.
And another finalist? Where's another finalist? Story, name?
About the future of solar power in Costa Rica, and my name's Connor Lignac from Elon University.
Connor from Elon. Another finalist? Where's another finalist? Finalist, name, school, story?
I did the life of water bottle. My name is Nancy Etcher from Akron.
The life of water bottle, okay. Another finalist? Yeah.
Olivia Urbanski, I'm from Loyola University, Chicago, and the last one was the white rhino. Male.
The last white rhino. Another story over here. Finalist, yes?
I heard about using artificial wetlands to clean off pharmaceuticals from wastewater.
Perfect. School name, story, school name.
Oh, Swanee, Vanessa Ross.
Everybody know where Swanee is? What state?
Tennessee.
What a crowd. All right. Is there another finalist I left out? I want to make sure yeah.
So my name is Navia. I'm from Berkeley, and I wrote a story about an interactive artist who uses his work to change minds about climate change.
Navia is speaking very softly, so I'm going to project. She's from Berkeley, and she wrote a story about artist who is?
Who uses his work to change ideas
Who uses his work to change ideas
about climate change.
about climate change. So we'll share all of this in a few minutes. Right now, I'm going to turn it over to my friend, colleague, Imani Cheers, to get us rolling. Imani, it's all yours.
Morning. How's everybody doing today? Good. Well, I'm beyond excited about our presentation this morning, and I hope you are too. Anand Varma. Who has seen Beauty and the Beast? Okay. How about Beauty and the Bizarre? Anand Varma, one of his biggest discoveries is figuring out how to make us imagine. He's a National Geographic explorer. He's a recipient of the Young Explorers Grant, which you'll hear about shortly. He's a scientist, but he's also a storyteller, using high-speed photography to take a look at parasites, bats, birds, and insects. Anand has been embracing the power of story in several different ways to demonstrate science, scientific discovery. He also has a TED Talk that's been viewed over 2.5 million times. Without further ado, Anand Varma.
Thanks, Amani. So before I really show any of the work that I've done for National Geographic, I want to tell you a story about how I became known as the gross bug guy at National Geographic. And as soon as I can get that photo up. So I'm not going to tell you about hummingbirds yet. I'm going to tell you about those at the end. This is how I became a photographer. I was a biology student at Berkeley. I had a plan on becoming a marine biologist. I was focused on a track in academia. And then I got a phone call at the end of my sophomore year at Berkeley. My instructor emailed me and said, this photographer's looking for an assistant. I see you with your camera on these field trips. Maybe this would be an interesting opportunity for you. I called this guy up. His name is David Lichwager. And my first job helping him was to rappel into these caves in Southern California and look for these cave-dwelling bugs for him to photograph for a story for National Geographic. And I had so much fun in this first project, I stuck with him for another 7 years. And I worked on projects at the top of giant sequoia trees, also looking for the insect biodiversity that lives up there. And we went up to the Arctic to do a story about ocean acidification. In all, I helped them with about 10 stories for National Geographic magazine. And so along the way, I started realizing all of the things that I really wanted to do in academia and science was to be out in the world, exploring and learning about the natural world and sharing that with other people. And I started realizing I could do all of those things through photography. So in between these projects of jetting around the world, I'd come back to Berkeley and I'd follow my friends in the biology department, like Vance Breedenberg, who was studying chytrid fungus in mountain yellow-legged frogs. So this is a deadly fungus that has been killing amphibians worldwide. And he figured out this way of preventing this infection. He discovered this bacteria that grows on some of the frog's skin. And he designed this experiment where he put the frogs in little baths in these Tupperware containers and soaked them in this bacteria to prevent them from getting this fungal infection. And so I would do these independent projects, kind of following my friends around, and I would work as an assistant. And there came a point where the assistant work kind of slowed down a little bit, and I realized I'm getting an amazing education as an assistant, but I am not yet ready to make it in the world as an independent photographer. And I need to go out and show my own work and figure out how to prove my own skills. And that's when I heard about National Geographic's Young Explorer Grant. And right now, the name has changed to Early Career Grant. And so I proposed a story to go to Patagonia and photograph the biodiversity of these wetland ecosystems. So this is one of the creatures that lives on the edges of these wet meadows. It's called the colonial tuco-tuco. And it's something that one of my professors at Berkeley studies, Eileen Lacey. And so she helped me design this proposal, and she helped connect me to local biologists and ranchers so that I could go down and have contacts and be able to travel through the region and find creatures like this to photograph. And one of the really valuable aspects of this grant, this opportunity, was, I had learned a lot about photography as an assistant. I had gotten a lot of chance to practice that at Berkeley with my friends, but I never really had been given the responsibility to follow through on a project on my own. And so learning how to plan logistics, find collaborators, and most importantly, actually prioritize my time in the field and figure out, okay, I have way more work here than I have time for. How do I choose what to do? This is an opportunity that gave me the chance to try those things in kind of a low stress, low consequence environment. I mean, if I failed, then nobody would really know about it. Whereas if I came back with interesting photos, it opened the door for me at National Geographic. Because I was a grantee, I could come here to DC, I could present my work to editors at National Geographic, and that started conversations. And because they never published these photos in the magazine, but they started asking me about, okay, what other stories do you want to tell? What projects are you interested in? So the first project I proposed was a story about parasites. And I was lucky enough that National Geographic already wanted to do this story, and so they decided to take a chance on me. Challenge is, the particular kind of parasites I wanted to photograph, these are the kinds of pictures that I would find online. And it turns out there's a really fascinating story about this interaction between a cricket and this worm. But I recognize, there is a serious challenge ahead of me here. Because as fascinating as the science is here, I'm going to have to get people to get over their visceral aversion to parasites and pay attention long enough to actually learn this. So I'm going to have to take a new approach to photography, something fresh. It's got to be interesting enough for them to learn about these creatures. So I thought, okay, this is how I had learned to take macro photographs up to this point. Clean, simple, sharp. I thought maybe if I could just make it sharp enough and clean enough, this would work. But it's not a bad photograph, but it's not interesting enough to really capture anybody's imagination. So I thought, okay, what else can I do here? I tried making it a little weird. But I think maybe I went too weird. This is a little bit too abstract. If you don't know what you're looking at here, there's nothing really to grasp onto. So what's important about this relationship is it happens in water. And so I thought, okay, maybe if I photograph this cricket and the worm in water, that's going to make more sense. It started to make more sense in my head, but it's still not an interesting photograph. So actually, I have a video of how I got the final picture that was published in National Geographic. This is a cage of infected crickets that's in my kitchen in Berkeley. And so I had this scientist, Ben Hanelt, FedEx me all of these crickets. He studies them in New Mexico. And every night I'd shake out the crickets and I'd inspect them. You can actually tell when the worms are ready to emerge. You'll see their coiled bodies darken in the belly of these worms. And I set up my little photo studio in a shed in my backyard. Probably should have gotten a haircut before I asked my friend to film me in my process. And so then I set up these lights. I put this cricket in the refrigerator to cool it down. It slows down the cricket, it slows down the worm. It means that when I set up this photograph in a little tray of water, both are moving slowly enough that I can compose the final image. And what happens here is this worm in nature, when it's ready to emerge, it forces the cricket to find water and drown itself so that it can emerge and finish the aquatic stage of its life cycle. So this is the final picture. And as you can see, this doesn't really look like a normal natural history photograph. I'm trying to create kind of a magical world that makes these creatures not just look gross, but seem interesting, seem mysterious, seem worth getting to know a little bit better. If you're wondering how I came up with this particular aesthetic, this lighting scheme, it came from a combination of looking at film noir, graphic novels, and Japanese animation. If you're wondering what Sailor Moon has to do with mind-controlling parasites, let me go through this again. Hard lighting, dramatic backgrounds, selective attention to detail. So what anime artists have figured out is that not all details are equal. So they put way more emphasis on her eyes and the reflections and the details because they recognized her eyes are a much more powerful way to convey emotion and to convey action than the pores on her skin or the texture of her hair. And so I took that idea and I thought, okay, what in these pictures do I really want you to pay attention to, and what's going to get in the way of telling this story? So let me show you a few more photos from this story. This is a crab I photographed in Armin Couris's lab in Santa Barbara that's been infected by a parasitic barnacle. So what this tiny little barnacle does is it crawls into the body of that crab. If that crab is male, it gives it a sex change. It turns them all into females, which means they grow a female anatomy. The critical part of that is an egg chamber that this parasite lays its own eggs inside and then activates the maternal care instinct of this feminized crab, so the crab cares for the eggs of the parasite. And every 2 weeks, tens of thousands of baby parasitic barnacle larvae hatch out. So all these tiny little specks you see, those are newly hatched parasitic barnacle larvae. And I lit this to draw your eyes to those tiny little specks, because I don't really care about the texture of this crab, the color of that crab. Those are not relevant details to the story that I want to tell. This is a frog that's been infected by a flatworm that forces it to grow extra legs. And that's because the flatworm actually wants to end its life cycle inside the gut of a heron. And so it uses this frog as bait by disabling it. That frog can no longer escape. It can't swim as efficiently. It can't jump as efficiently. So it's more likely to get eaten. And this tiny little parasite is just using this frog as transportation. This is a ladybug standing guard over the cocoon of a parasitic wasp. So the wasp, the adult wasp, lays a single egg into this ladybug. The egg hatches, and that larva feeds on the insides of the ladybug. But it knows how to keep it alive. It knows how to avoid its vital organs in order to not kill it. And then it squeezes its body out of the ladybug, and it spins a cocoon. And because it's vulnerable in that cocoon stage, it can't run away, it's busy developing into an adult, it has figured out how to force the ladybug to stand guard and protect it from other predators. And this is the photo that eventually made it to the cover, which is kind of cool because bugs don't make it on the cover of National Geographic very often. This is another example of one of these bodyguard relationships. This is another wasp that's infected this caterpillar, and it's forced the caterpillar to guard the cocoons. And I actually filmed this whole life cycle in Jeff Harvey's lab in the Netherlands. I'm going to show you that. I'm just going to warn you, it gets pretty gross, but I think it's cool. This is a wasp laying several dozen eggs into a newly hatched caterpillar. So that caterpillar survives the sting. It lives a relatively normal life for the next week or so, feeding, growing, until those wasp larvae are ready to emerge, at which point they chew their way through the skin of the caterpillar, and they emerge by the dozens. What they're doing, they're actually spitting their cocoon as they're pushing their way through the body of that caterpillar. What's remarkable about this process is the caterpillar survives all of these creatures emerging from its body. It then crawls on top of that pile of cocoons, it spins an additional layer of protective silk, and then it takes its post as a bodyguard, and it'll whip its head back and forth anytime it's disturbed. And the whole purpose of that complex interaction is to prevent this, which is a hyperparasitoid wasp, which specializes on hunting the parasitic wasp that just came out of the caterpillar. So an additional layer of complexity. This is an ant infected with an Ophiocordyceps fungus. So this is a fungus that gets into the body of the ant, and then it forces it off the forest floor to the end of a leaf, and it forces it to fix itself there, to grab on. Then kills the ant, consumes its body, and then sends these reproductive stalks, like antlers, through the exoskeleton of the ant. And then it releases its reproductive spores. And because the ant is off the ground, those spores can travel further through the forest to more efficiently infect more ants. And I was working with João Araujo, a graduate student in Brazil, who found this specimen, and he actually found an ant that was in the process of this manipulation. So I took a video. This is the last few hours of this ant's life. The fungus has forced it to bite down the edge of this leaf, and then it kills it there. So I took a time lapse of the first night, and you can see the fungal tissue bursting through those weak parts of the ant's exoskeleton. And then over the course of the next week, it sends that reproductive stalk out of the back of the ant's head. And this fungus looks different than the photograph I just showed you because it's a different species. It attacks a different kind of ant. There are actually hundreds of different kinds of this fungus that attack different species of insects. And there are thousands of different species of these host-manipulating parasites. When you look at parasites more broadly, they estimate between 30 and 50% of the animal biodiversity on our planet is parasites. And so when you think about needing to protect biodiversity on the planet, we have to actually include parasites because they're a huge part of that. So this I think, is the queen of all mind-controlling parasites. It's a emerald cockroach wasp. It's a wasp that hunts cockroaches to feed its young. But it's much more sophisticated than any other predator that I know. The way it hunts this creature, which is bigger than it, and it's heavier, it can't overpower it, it starts by stinging it right behind its head. That paralyzes it temporarily, and then it weaves its stinger into the brain of this cockroach. And its stinger has these special feelers that can detect the part of the cockroach's brain that is responsible for the motivation for movement. And it injects a special venom cocktail that just disables that part of the brain. What that means is this cockroach, all of its muscles still work, its body still works, but the wasp venom breaks the connection between the part of the brain that can generate movement. So it can no longer run away. It can no longer decide to run away. Instead, what happens is the wasp grabs the antenna of the cockroach. And there's some pathway still intact between that antenna and the movement of this animal. So by tugging on the antenna, the wasp can actually walk the cockroach like a dog on a leash back to its burrow where it buries it alive with a single egg. And that egg hatches and consumes that cockroach. So you might wonder, okay, why should we care about these gross little bugs? Like, surely there are more important things to do than studying random little wasps. Well, consider this. What happens to that cockroach is actually very similar to what happens to Parkinson's patients. Their muscles still work, but the brain loses its ability to control those muscles. And the mechanism is similar enough that Frederick Lieberstadt, who's the Israeli scientist who studies this wasp venom, is actually collaborating with Parkinson's researchers to develop better treatments for Parkinson's by studying the action of this wasp venom. So I think that's why I think it's important to tell science stories and why it's important to do basic research, because you never know when you're going to find that you have something in common with a cockroach. It's not obvious at the start. So for each of these stories, I have to take a slightly different approach to figure out the lighting on the fly. And so each of these photographs, I set up a time-lapse camera to show my process. So here are a couple of those videos that show me at work. This is a hotel room in Costa Rica where I'm photographing a spider, and I have to hide the spiders in these white bins so that the housekeeping staff doesn't throw them out. This is a hotel room in South Dakota. And really, as you can see, about 90% of my field work is just rearranging hotel room furniture. This is a lab where Fanny Morey studied that ladybug. And so they gave me a lot of lab space to spread out to try my photography experiments. And this is in Colorado, where they study the deformed frogs. So halfway through this project, I needed to send an update to my editors at National Geographic. They wanted to see my best photos up to that point. I was behind schedule, I was behind my deadline for turning in these photos, and so I was staying up late at night, and I was listening to this techno music to kind of just keep myself awake. This weird thing happened where the beats of the music and the patterns of the images as I was scrolling through them on my screen synchronized for just a little bit. And as a joke, I made this stop motion video by exporting all of my photographs, playing them back at 15 frames a second, and setting them to this music. So I'm going to show you a shortened version of the dubstep parasite music video I made to show my editor my photographic process. Here you go. Thanks. So at the end of this story, National Geographic asked me to photograph a story about honeybees. So I decided the first part of this process was I should get some bees in my backyard. So this is Alice Rosenthal. She's a beekeeper in Oakland who came and showed me how to keep bees. She lent me a beehive. She showed me how to handle them, how to care for them, how to look for problems. And I thought, okay, if I have bees in my backyard, maybe I'll have enough time to get to know them intimately, to have ideas about how to photograph them in a new way. And really, when National Geographic asked me to do this story, at first I was not that excited about it. I didn't really want to do this story. I thought, what can I possibly say or show about honeybees that has not already been said? You know, what use is there to have one more story about colony collapse? But at the same time, who am I to say no to National Geographic when they're offering me a story? So I thought, okay, well, let me take what I learned from parasites and try to apply it to bees. And so I would sit in my backyard with this little beehive, and I'd try to photograph bees the way I'd photograph those parasites, with the little fiber optic lights, trying to backlight them. I photograph them in flowers. This is an almond flower. Taking close-up portraits of their faces. And you know, again, it's not a bad picture, but it's not so different than all the pictures that I'd already seen. And I thought, if I don't have something new to say about this story, and if I don't have something new to show about this story, what's the point of doing it? But at the same time, I was happy to just be trundling along in my backyard. The other reason I kept bees in my backyard is this is a story that probably I haven't told at National Geographic, but my girlfriend had just moved in with me at this point, and I just spent 8 months of the year traveling for parasites, and now I had a way to do a story in my backyard. And so I thought, if I don't have to travel and I can do this whole story at home, maybe she won't break up with me. Which, you know, I think that worked out in the short term, but long term, not so much. So I was kind of happy. I was happy to just keep trundling along and, you know, work on the bees for a little while. But I never got past the stage of like a decent macro photo. And 8 months into this story, you know, my editor, Todd, calls me and he's like, you know, we're getting a little worried about your progress on this story. And we need to see some new direction from you, ideally in the next week. And I thought, this was my first real panic moment. I thought, whoa, I really have the chance to screw this up. Because if I don't find some new way to visualize these creatures, I think they're going to cancel the story, and I don't know if I will ever work for them again. So I spent, at this point, it was helpful that I had become kind of intimate with the life of these bees, but so I could figure out how to come at it with a new way, figure out some way that looks unusual, that looks fresh. And as I was looking at one of these frames of a beehive, I noticed one that had gotten stuck as it was trying to emerge, as it was kind of crawling out of its cell for the first time. And I thought, okay, maybe this is not so much about portraits of individual bees, but showing it in context might make it look a little bit different than what I'd seen before. So this is a fresh young bee just as it's emerging from its brood cell. And I use this technique called focus stacking. This is actually 24 images taken a few millimeters apart, a few microns apart actually. And then the in-focus elements of each of those photographs is combined to show you all the hairs on this bee's face. And so this was enough to at least convince National Geographic to back off a little bit and give me more time and to introduce this story. But I also recognized, you know what, I can't really tell this story just with interesting macro pictures. So I decided to visit labs across the country and show how scientists are studying the problems that bees face. So for example, Frank Rinkovich at Louisiana State designed an experiment to look at how pesticides affect the health of bees. The way he designs this experiment, it's pretty simple. He puts them in little paper cups. He pokes holes in the bottom of the paper cup, and he sends carbon dioxide into the cup where the bees are, just to temporarily sedate them. And when they're not moving, he can dose them with a careful droplet of pesticide, in this case, a mosquito control pesticide that they wanted to see if it was having a side effect on bees. But this is not necessarily the most sophisticated way of testing the effects of pesticides. It's a bit of a crude way that assumes that bees are getting a very high dose. Chris Mullen at Penn State actually is using a method that looks at really subtle effects of pesticides on bees. And the way that this experiment works, they put the bees in a little plastic cone to restrict their movement. Then he exposes them to scented air, like a floral smell. And after that smell, he gives them a Q-tip dipped in sugar that they can lick as a reward. And then you wait a while, and you give them the same smell. If they stick out their tongue, you know they've learned to associate the smell with the reward. It's like Pavlov's dog. It's a way of measuring a bee's memory or its learning ability. So what you can do is you can measure that for a bunch of healthy bees, and then you can expose them to a really tiny amount of pesticide that's more realistic for what they would be exposed to in the wild, and see how they respond to that. And what this lab shows is that pesticides at levels much lower than what would kill them outright are affecting their behavior, affecting their ability to remember where their food is. And ingredients in agricultural sprays that were thought to be safe for bees are actually having these subtle, sublethal effects. But as much concern as even Chris Mullen has about pesticides, he and every other bee researcher I talked to was much more concerned with the Varroa mite. So this is a parasite about 2 millimeters across that crawls into the beehives, it crawls into where the baby bees are developing, and it chews on them, it sucks their blood. It doesn't kill them outright, but those bees that have been infected with this mite, they grow up a little bit smaller, a little bit more deformed, and with a weakened immune system. What that means is they're more susceptible to fungus, bacteria, viruses, and they have a harder time detoxifying pesticides because their immune system is compromised. So this one parasite amplifies every other stress factor that bees face. So one of the ways that scientists are trying to manage this problem is through an artificial breeding program. This is at the USDA Bee Lab in Baton Rouge, where they identified certain bees that actually have a natural ability to fight mites. And they designed a breeding program to select for that trait. So this is a queen bee. They glue a little number on her back to keep track of her in the hive. And this queen has this trait, and so they're using her in this breeding program. Here she's surrounded by nurse bees. Those are bees in the hive that follow her around and clean her and feed her. And you can see she's actually getting a meal. Her tongue is sticking out, and one of those worker bees is giving her a meal. And this is how you breed bees in a lab. This is Shannon O'Brien's hands. She's the lab technician there. They use artificial insemination to select for certain bee traits. And so they sedate the queen bee in this little plastic tube using carbon dioxide, and then they inject semen into her oviduct with this precise instrument. So they bred these mite-resistant bees in a lab, and then they partnered with Brett A.D., who, with his brother, runs the largest commercial beekeeping operation in the world. So this is him opening one of his 72,000 beehives. So what the USDA did is they took these mite-resistant bees in the lab and they integrated them into Brett's operation. And Brett moves these bees from South Dakota to Texas to California to Washington on this migratory pollinating operation. So year after year, the USDA can go back and see which of these bees that are mite-resistant have thrived in this migratory operation, are still producing a lot of honey, that are able to survive winter, with the long-term idea that they can produce mite-resistant bees that are still attractive to commercial beekeepers, that have all these other traits they're looking for. So back home, I still have this hive of bees in my backyard. And I've noticed, there's a glass pane. It's called an observation hive. I can look inside of the hive and watch them crawling around and going about their lives. And I noticed, there's little cells in the hive where the egg is being laid. You can see the little larva spinning around. And then there's the cocoon is covered in wax. You don't really get to see what happens between the larval stage and the adult. But something magical happens there because they start out as this white grubby thing and they come out as this furry winged creature. I thought, this is happening right in front of me. I want to figure out how to show this with a camera. So Billy Sink was a researcher, a lab technician at UC Davis, actually helped me figure out how to raise bees inside of an incubator. And so you could remove the little wax cap and provide an environment that was appropriate and safe for the developing bee inside, and actually show that using time-lapse imagery. So what I'm going to show you next is the first 21 days of a bee's life condensed into 60 seconds. This is a bee egg as it hatches into a larva. That larva then spins around itself, feeding on this white secretion that nurse bees are feeding on. The larvae then transform into pupa. That's the stage in their process where their head and their legs start to separate. Here's that same pupation process seen from above. You can actually see a Varroa mite running around in the cells there. This is where the tissue starts to differentiate and the pigment in their eyes starts to darken. The last step of the process is their skin shrivels up and they sprout hair. The last story I'm going to share with you today is on hummingbirds. In a lot of ways, this is this project I've worked on the longest. So about 11 years ago, I started working with a friend of mine who was a graduate student at Berkeley who studies hummingbirds. This is Allen's hummingbird in Berkeley that he was studying. And I would travel with him. He included me on his grants, and so I would travel through Central and South America, and I'd photograph the different species we came across, like this white-eared hummingbird in Guatemala, and this volcano hummingbird and scintillant hummingbird in Costa Rica. And at first, my goal was to just make these portraits of these different species. But at a certain point, I realized, okay, this is, again, these are nice photographs, but they're very similar to the ones I've already seen. So I want to figure out how to show these creatures in a new way. The project that I proposed to National Geographic was to show how scientists study hummingbirds. So for example, this is Chris Clark weighing a Cuban bee hummingbird. And hummingbirds have this interesting behavior that when you flip them upside down, they get disoriented, and they hold perfectly still. And so he's using that to measure this hummingbird. And you can see on the scale, that's 1.83 grams. That makes it the smallest hummingbird in the world, smallest bird in the world. This is another way to see its scale. I trained one to perch on a penny, and that penny is 2.5 grams. So Chris is taking all of this data out in the field, and back in the lab, he's using a wind tunnel to measure its flight performance. And so I went to different labs that are using different kinds of scientific tools to answer questions about hummingbirds. This is a lab at Stanford where Rivers Ingersoll built a contraption to measure the force of a hummingbird that a hummingbird generates in flight. This is an experiment that Benny Gollard designed in Doug Altro's lab in British Columbia, where they play optical illusions on the side of a hummingbird cage in order to decode how the hummingbird uses vision to balance in the air. This is how they study the aerodynamics of hummingbird flight. They use a tiny fog machine to visualize the airflow around a hummingbird. This is what hummingbird tongue looks like. It's a forked tube that can pump nectar into its mouth. And this is how a hummingbird dries itself off. It shakes itself so vigorously those water droplets shoot off of its body. This was research done by Victor Ortega Jimenez at Berkeley. So this is my job, essentially. It's trying to use photographs to tell weird, complicated, and sometimes unpleasant science stories. And I can't always tell what people are going to respond to, especially with a larger audience. And so I'm trying to mix it up between gross bug videos and sad bee photos and silly time lapses, essentially plucking different emotional strings to try to make sure people don't tune out. And I'm trying to find the right balance between familiar and novel so that people have some context to absorb new information. And in my opinion, I think the most basic way you could define storytelling is providing information in a structure that makes it more relatable. And the simplest way you could define my work is I'm trying to get people to redefine how they relate to the natural world by sparking a sense of wonder. With that thought in mind, I have one last video to show that shows the movement of hummingbirds 100 times slower than what your eyes can see. Here you go. Thank you. So I can put up a little bit of contact info if you want. I have questions for later on, but I'm happy to take some questions. Could we get the house lights up a little bit?
Ladies and gentlemen, Anand Varma. One more time.
Thank you. Thank you.
Wow, that was so much. I wish I would have seen that yesterday so I wouldn't have spent the whole time with my mouth gaped open, staring in wonder. One of the, we're gonna take a couple questions, but I have one or two for you
Sure.
before we open it up. So please, if were in awe like I was, hopefully you had a moment to jot something down or tweet something and get those questions ready. Well, first of all, I'm really excited that Nat Geo gave you more time on that bee story. So way to kind of procrastinate enough to get to like where you're going. But one of the things that I noticed in your conversation this morning is that you are looking at the wonder in nature that we don't see so clearly with the naked eye. Can you talk a little bit about your process in developing stories? Because I don't know if I would have been, I don't know if I would have thought about parasites and how there's beauty in parasites coming out of the back of crickets. Like that just wouldn't have been a story idea. So where do your story ideas come from?
My story ideas come from essentially my friends who are still working in science. I don't really consider myself a scientist now. I'm not really doing research.
Right.
My work is communicating the work of others, scientists. And so the parasite story came from a friend of mine at Berkeley, a classmate who went on to do her PhD on parasites. Oh. And she called me one day, and we were just catching up, and she said, you really got to do this story about these crazy mind-controlling parasites. And she gave me a couple of examples. The crab was one that was her lab studied. Okay. And so, you know, the hummingbirds, that was another. It started with a friendship with a biologist who's doing work that I thought was fascinating, and I followed him around. And, you know, the early stages, the for the first years of working at National Geographic, there was not really like a coherent strategy or plan or how I choose stories. Just kind of try to impress the editors whichever way I can.
I think that's successful.
Well, yeah, no, it's worked out so far, but when I think about, okay, well, what ties all this together? What is it that I'm responding to? What is it that I'm thinking about for future stories? I think the thing that ties it all together is what you mentioned. It's making the invisible visible.
Yeah.
And with parasites, it was things that are too small to see. With bees, in the case of the time lapse, it was a process that was too slow. Hummingbirds is too fast. So it's not just about little stuff, but it's, how can I use a camera as a way to enhance our own vision?
And understanding.
And understanding.
Yeah.
And I think about that in a way that's, what's most interesting to me is what's right at the cusp of our perception. Mm-hmm. Something like that, the hairs on a bee's face. Something you don't notice, it's too small to notice,
yeah.
But I'm hoping with the way that I photograph it, it's something that maybe you'll look at a bee differently.
Absolutely.
So it's at the edge so that what I want you to do is to go back and think about the iridescence of a hummingbird's feather that you can see if you pay attention, sure. But you may not have noticed if you didn't know it existed.
Or the fork of the tongue. I thought that was quite
yeah.
beautiful and amazing. And I love how you're adding, I tell my students all the time, okay, we're not here creating music videos. However, I don't know if that was your intention, but you created some pretty amazing music videos. Can you talk about how you put together your time lapse and how you're using powerful audio to enhance the experience through the visuals?
The audio, in my opinion, is the thing that I want to develop more, that I feel like is my greatest weakness. Oh. The dubstep thing was a total.
I guess he has to have one, so we'll take it. That's where we're at.
No,
okay.
I have lots, but that's the one that relates to this particular question.
Absolutely. Okay.
The dubstep thing was total luck. That only came together because this thing happened, and it never would have happened if it hadn't been that moment.
Trying to stay awake. And
yeah, and I recognized with that how important music is mm-hmm. in this sort of emotional response of the content. And so
there were some pretty graphic photos in there, but with the music it didn't feel gross.
And that's what I'm trying to learn more about and to I feel like I stumbled into it and now I'm trying to decode it from nice. Backwards. Like, okay, wait, why did that work? How can I intentionally get that to work in the future as opposed to accidentally get it to work? Which is really the way that my photo process works too. You sort of bumble along, you take pictures of random stuff, and one day you're like, wow, I got this picture. I have no idea how I got that. And the process of becoming a more established and more successful photographer is figuring out how to do that more intentionally. And so I want to figure out how to get the music part and the visual, the video part better because
nice.
I'm kind of just bumbling along.
Well, it's working so far, so keep bumbling. I think we have our runners available with our microphones. So we'll take about 2 to 3 questions from the audience. I saw your hand up really quickly. If you could just introduce yourself for us, that'd be great.
Hi, my name is Elaine. I'm from Yale NUS, and I just want to say I'm fascinated by your fascination for these parasites. And it's just amazing that these parasites seem to know how to hack nature's designs. Like, how does a flatworm know that getting into a frog is going to take it to a crane's belly? That's amazing. So you talked a little bit about the applications. You mentioned about how with the cockroach, this is similar to what we see in Parkinson's patients. So in kind of like getting into the nitty-gritty of all these creatures, what are the other applications that you have seen from this?
That's a good question.
Yeah, I think the hummingbird side, there's a lot of kind of inspiration at looking at their flight. This is a tiny little creature, a couple grams, that's able to maneuver better than any of our machines or robots. And so there's, efforts to try to better understand how to fly at that scale. Another thing that's interesting about hummingbirds is not just flight, but their hearts are beating at 1,000 times a minute, and their wings are beating at 100 times a second. And so physiologically, they're operating at the extreme end of all vertebrates. So thinking about, okay, how are they dealing with all these kind of metabolic challenges. It's a way of, I mean, that's kind of one extreme, and then you're looking at these parasites that are sort of breaking all these rules that I kind of thought I knew as a biology student. And so all of these are kind of, there's sort of these waypoints that sort of show what is possible in evolution and what is possible in physiology. And so I think there's certainly much more specific examples that I'm not aware of as somebody who kind of just drops into a field and scrapes the surface of it. But my general sense is that all of these things are sort of expanding how we think about, or how I think about how evolution works and how ecology works.
Nice. Biodiversity. Awesome. Any other questions from the audience?
I'm here.
Yes. Hello.
Sorry, I've got myself pinned in here with one of these desks. Zach Smith, SUNY ESF, and in storytelling, one of my passions is podcasts. So as I'm watching this video, I'm like, man, has anybody got a microphone to figure out and to find the sound of, you know, the tongue coming out to bring people even closer to this experience? And I can think of all the complications that would entail, including your ears to having to magnify those small sounds and filter out the bigger sounds. But is anybody doing that? That you know of?
Good question.
Not off the top of my head. I've kind of had this tunnel vision of visual media, and I'm kind of now trying to figure out, okay, how do I collaborate with people who have been thinking about audio or narrative in different ways. And so I don't know off the top of my head, but I think there's a lot that cool stuff that could be done in that space.
Awesome. Thank you. Thank you. And just introducing anyone else. So is there anything that you can share about projects you'd like to work on next?
Sure, yeah. So I haven't really started the next project, but I think it's going to be on jellyfish. Ooh. And the way that I think about jellyfish...
Everyone got really excited. Like, that sounds amazing. Okay.
I think there's like an interesting take on showing the invisible there, too. It's not small or fast.
I got stung scuba diving a couple months ago. Didn't see it, felt it.
Yeah.
I felt it.
So they're transparent. And
Absolutely.
there's techniques, kind of borrowing the optical design of microscope condenser lenses. You can scale that up, and you can increase the contrast, and you can visualize all these interesting musculature and tissue inside of the jellies. And so just that alone, and then the life cycle of jellies,
Yes.
thinking about showing a jellyfish being born, it's a weird life cycle.
Okay.
You don't really think,
I'm
it's
excited.
not obvious. So
I go diving. That might be our next Story Fest I don't know, but we need to talk about that.
Give me
sounds
like
amazing.
2 years, I'll figure this out.
All right, we'll be there. We'll be there waiting. That sounds amazing. Thank you very much. Thank you so much.
Sure.
We really appreciate it.
Thank you.
Thank you. Don't leave just yet, though.
Are we bringing up Dustin?
Yes, absolutely.
Yeah, great.
Yes. Right? I know, right? Round of applause. Stand up. Okay, so we learned that you received a Young Explorers grant.
Early career grant.
Early career grant is what it's called now. Yes, Young Explorers Grant now. And I'm sure many of you were curious about what that is, how can you get more information. So we decided to make it easy for you, okay? And we brought Mr. Dustin Renwick from Nat Geo to give you a little more information about that program. So everybody take notes, get ready. I know you have some amazing ideas, and we're gonna learn how you can apply, where you can get more information. So please welcome Dustin. How are you?
Thanks.
So please share.
Are we on now? Yep.
Yes, you are.
Hi, everybody. I'll make this quick because I'm also going to be upstairs after this for the meet and greet and kind of networking.
Absolutely.
So it was called Young Explorers. It's now called Early Career Grant. But it's part of what the National Geographic Society does in giving grants to emerging educators, conservationists, scientists, and storytellers.
Awesome.
So that was a storytelling grant, what it was then. It's the same basic principle is that we're looking to support the best and the brightest and really help them launch their careers. And if you heard what he was talking about earlier, it's less about the actual project, but I think some of the conversations that you were able to have. Mm-hmm. This is the door into National Geographic. And it's about those conversations with editors, the conversations with other early career grantees that you might have. We're really looking to build the ecosystem. So the quick thumbnail sketch is that they're 5 to 10K,
Okay.
about a year-long projects. They can
Are
be.
there age requirements?
You have to be at least 18.
Okay.
So anybody who's in college is probably going to fall into that category.
Absolutely.
And then there's no maximum age limit, but basically we say if you've been working professionally for 5 years, you've probably graduated to the next class. So happy to talk more. I'll be around upstairs again. I can sort of consult on ideas. I see all the applications when they come in.
Are there any deadlines coming up that people need to be aware of?
Yeah, so we grant quarterly.
Okay.
So we just passed the spring one. So the next quarter, the next deadline will be July 10th.
Ooh, plenty of time. Yeah,
plenty of time to
absolutely.
think about. And again, what I tell people is, we grant quarterly, so don't feel like you have to rush to this next one.
Yeah.
Take time, think about a good idea, because it is competitive, but it is the door into National Geographic and can open up some really interesting opportunities. So.
Thank you, Dustin. You'll
Absolutely.
see Dustin upstairs. Thank you, Anand. Thank you all. And I'm going to turn it back over to Frank Sesno. So thanks.
Yeah,
Oh, we got one more? I'm sorry.
I just want to reiterate the
Yes.
thing you said about the ecosystem. That is the thing that I valued the most, is being able to meet all these other young explorers, our early career grantees, and getting sucked into this system. Whether you get a magazine story out of it or not, that's been the most amazing thing, is all the other cool people I've gotten to meet, and the effort that National Geographic puts into building those opportunities for you. It's not just, here's some money, go figure it out. It's like, now that you've proven yourself, come in
What else can you do, right?
Yeah, and let's help you do the next step. And that's something that I think is really special.
So should we do one selfie with us, with them in the background before we, all right, so everybody turn around. Here we go. All right, here we go. Anand, get in there. All right, that's going up.
Thank you.
Is this amazing?
All right.
Unbelievable? Like, really amazing? I've been talking about compelling characters. How do you make, like, a bug compelling? You did it. And you did it both with your picture, and did you hear how he told the story around the picture? That's what we mean. Storytelling isn't just once upon a time or a video piece. It's the way you think about something and the way you communicate that out. And that's the magic. Thank you so much. Thank you. You're going to hang around for a couple minutes?
Yep.
And
I'll be upstairs,
people can mix and mingle. All right,
Yeah.
great.